Hello, my name is David Merrill. I'm a PhD student at the University of Wisconsin. I'm also with the Morgridge Institute of Research. Today I'm presenting work entitled Inferring Signaling Pathways with Probabilistic Programming. So some motivation to start with. We're trying to solve a problem called signaling pathway inference. Here's the idea. Cells regulate themselves via complicated biochemical processes called signaling pathways. You can think of a signaling pathway as a directed graph where the nodes are proteins and the edges are regulatory relationships between them. We want to infer signaling pathways from time series phosphorylation data. In these slides, I'll use the variables x and g to refer to time series data and graph structure, respectively. So that's our problem. How do we solve it? We formulate the problem in terms of dynamic Bayesian network structure estimation. And we take a Bayesian approach. Namely, we want a posterior distribution over graph structures conditioned on time series data. We decompose this posterior into a graph likelihood function and a graph prior distribution. We admit prior knowledge in the form of a weighted directed graph. This could be derived from signaling pathway databases, such as KEGG, for example. For our graph likelihood function, we use a dynamic Bayesian network. A dynamic Bayesian network, or DBN for short, is a sequential probabilistic model. For our purposes, you can think of a variable in one time step as being a noisy function of its parents in the preceding time step. The structure of the DBN is governed by the graph structure. So that's how we connect the time series data to a graph structure. So the product of the likelihood and the prior gives us an unnormalized graph posterior distribution. So we have this posterior. Now we need a way to use it to compute probabilities. We need an algorithm. But to do that, we designed this method called sparse signaling pathway sampling, or SSPS for short. SSPS is a Markov chain Monte Carlo method. That is, it uses MCMC to sample from that posterior distribution. In other words, it goes for a random walk through the space of directed graphs. And that random walk is governed by a particular proposal distribution, which proposes moves through the space. Our biggest contribution in this work is that we invented this new efficient proposal distribution that acts on the parent set of a node. It proposes one of three moves. It might propose an add parent move, a remove parent move, or it might propose a swap parent move, which is just a simultaneous add and remove. And the proposal distribution assigns probabilities to those moves in a way that biases the exploration towards sparse networks. Intuitively, if the parent set is too small, then add parent moves are more likely. If the parent set is too large, then remove parent moves are preferred. And if the parent set is about the right size, then swap parent moves are given high probability. We implemented SSPS as a probabilistic program. We wrote it in Julia using the GEN probabilistic programming language. Probabilistic programming is a methodology for building probabilistic models. The main idea behind it is that if you have a generative process, you can think of it as a program written in a special programming language, which we call a probabilistic programming language. In our case, we used the GEN probabilistic programming language. You can find our full code base on GitHub. That includes SSPS's source code. It also includes a snake-make workflow for reproducing all of our results. So all of that sounds good, but we still haven't talked about whether SSPS actually works or not. In order to answer that question, we evaluated SSPS on simulated data and on experimental data. In our simulation study, We randomly generated true networks, used them to generate some time series data, and then made random modifications to the true networks in order to get corrupted prior knowledge. After we generated those simulated data sets, we would evaluate SSPS's ability to predict the true edges from the corrupted prior knowledge and the time series data. And we also compared SSPS against other established methods in this simulation task. The results are summarized in these heat maps. Long story short, this top row are SSPS's results. The other rows are for other methods. Blue is good, red is bad. SSPS got a lot of blue. And so that's good news for SSPS. The takeaway here is that SSPS did a good job of recovering true edges, and it scaled to larger problems than the most closely related DBN-based methods. So that was the simulation study. We also evaluated SSPS on experimental data from a dream challenge from a few years back. The basic idea of the challenge is this. We had 32 biological contexts. In each context, we had a set of phosphorylation time series data. And the goal was to use that time series data to infer phosphosites downstream of mTOR. In each context, there were about 40 proteins being measured over time. We compared SSPS against a few established methods on this dream challenge. The results are shown in these bar charts. We have the 32 contexts here. The main takeaway from this is that SSPS attained similar performance to established methods. However, there were some issues in the design of the challenge, which made it difficult to say anything much stronger than that. So, in conclusion, there are some takeaway points that I would like to drive home here. First, SSPS infers signaling pathways by sampling dynamic Bayes net structures. SSPS improves on past Bayesian DBN methods by using a new, efficient proposal distribution that biases the exploration toward sparse networks. SSPS performs comparably to established techniques on our simulated and dream challenge tasks, but it demonstrates better efficiency and scalability than closely related Bayesian DBN methods. And lastly, SSPS demonstrates the potential of probabilistic programming to model complicated biological systems. We have a couple of references. I would like to acknowledge my advisor and co-author, Anthony Gitter. I would also like to acknowledge our funding from the NIH and the NSF. With that, I am finished, and I welcome your questions.